Good day, everyone. I am Angelica from Bachelor of Secondary Education, major in Sciences. And today, we will be delving a new topic which is entitled The Central Dogma of Molecular Biology. But before we will moving on through our further discussions for today, let's have first our learning objectives. So here's our learning objectives. At the end of this topic, you will be able to state the central dogma of molecular biology. Second, describe the structure of RNA and identify the three main types of RNA. And third, give an overview of transcription. And fourth, describe the genetic code. And lastly, explain how translation occurs. So about the central dogma of life, DNA dictates that the process or the structure of mRNA in a process known as transcription, and RNA dictates that the structure of protein in a process known as translation. So this is known as the central dogma of life, which holds true for all organisms. So the central dogma of molecular biology describes the flow of genetic information in cells from DNA to messenger RNA to proteins, and because the information stored in DNA is so central to the cellular function, and the cell keeps the DNA protected and copies the form of RNA. So according to Francis Crick in 1958, a discoverer of the structure of DNA, that is that the central dogma of molecular biology deals with detailed residue by residue transfer of sequential information, and it states that such information cannot be transferred back from protein to either protein or nucleic acid. And it was proposed in 1958 by Francis Crick, a discoverer of the structure of the DNA. In other words, protein information cannot flow back to nucleic acids, and fundamental framework to understanding of the transfer sequence of information between biopolymers. So let's have here the basics of about the cell organization between the prokaryotes and eukaryotes. So all cells, whether prokaryotic or eukaryotic rather, undergo this process. So in prokaryotes, organisms without a nuclear membrane, is that DNA undergoes replication and transcription, and RNA undergoes translation in an undivided compartment. All three processes are simultaneously occurs. On the other hand, in eukaryotes, organisms with a nuclear membrane, DNA undergoes replication and transcription in the nucleus, and proteins are made in the cytoplasm. RNA must therefore travel across the nuclear membrane before it undergoes translation, and this means that transcription and translation are physically separated. So the central dogma of molecular biology explains the flow of genetic information from DNA to RNA to make a functional product of proteins. So when we say proteins, proteins are the large molecules made of chains of amino acids, and proteins are essential to the functioning of our cells. The central dogma suggests that DNA contains the information needed to make all of our proteins, and that RNA is a messenger that carries this information to the ribosomes. So when we say ribosomes, they are the small organelles found in our cells. So they are responsible for assembling amino acids into proteins during protein synthesis. So the ribosomes serve as factories in the cell where the information is translated from a code into a functional product. So as you can see in the picture right here, is that ribosomes are like dots. As you can see, a green dots. These are called the ribosomes. And the process by which the DNA instructions are converted into a functional product is called the gene expression. So, when we say gene expression, RNA carries the code from DNA to ribosomes where proteins are made. So, let us consider here a light bulb and a switch. So, genes are the light bulb and the switch is the expression control. Now, when the light switch is in the on position, it means genes are expressed. But if the light switch is in the off position, it means genes are not expressed. So if certain genes are expressed rather, then certain proteins will be made. However, if the certain genes are not expressed because of the light switch off, then proteins will not be made. So there are other things that can influence gene expression. So environmental factors can influence what genes are turned and off or can mutate or alter DNA. So these factors can be internal or external. So by looking on the couple pictures here, they were hinting about a radiation of being an external. On the other hand, internal is being like a diet. So when the fetus, so here is the example of external factor, a exposure to alcohol while in the womb. So symptoms can include physical and mental abnormalities. So as you can see, this is called a fetal alcohol syndrome. So when the fetus is exposed in alcohol while in the womb of the mother, now this is not just a small amount of alcohol. This is the certain amount of alcohol that the fetus exposed to a certain symptoms that may include physical and mental abnormalities, just like what I've said a while ago. So you have seen a picture of a child who have felt a fetal alcohol syndrome because she was exposed of too much alcohol while still in the womb of her mother. So you can observe that the child have a small head, small nasal bridge, and these are the factors that which are affected by the gene expressed widely while the fetus is developing inside the womb of the mother. So this is another example for external factors. In sea turtles, gender is determined by the temperature of the sand during development. So it says that during warmer sand, there are some female turtles. On the other hand, in cooler sand, there are some male turtles. So we look on the internal factors. The great example of the hormones that express gene expression is puberty. So these hormones produced by the body can change the way that genes get turned on and off. So when you go through a puberty, hormones are released certain amounts and some more than the others, and they influence which genes express at what time and how the process of maturation goes about. So we have here a protein synthesis, or the major processes of protein synthesis. The first one is the transcription, in which from DNA make a new copy of messenger RNA. On the other hand, in translation, from messenger RNA will make a new copy of a proteins. So these are let's have first the differences between the DNA versus the RNA. So let's know about the difference between the two of them. Then we have here in the sugar, the deoxyribose for DNA and ribose for RNA. Also in nitrogen bases are slightly different. For DNA, we have here ATCG, and while in the RNA is AUCG, in which it has no thymine. So however, it replaced by letter U, which is called uracil, and the shape of the DNA is a double helix, while in RNA is only have single helix, and the DNA is located in the nucleus, while RNA is located in the both nucleus and cytoplasm. So here's the structure of DNA between the DNA and the RNA. Now, there are different types of RNA. These are the messenger RNA, which carries copies of DNA. Second, the transfer RNA or the tRNA, in which it transfers amino acids. And lastly, the ribosomal RNA, which is called also the rRNA, which makes up the ribosome. So here are the structures of types of RNA. As you can see, we have the first picture shows the messenger RNA. The second is the ribosomal RNA, and the third structure is the transfer RNA or tRNA. So in transcription, a section of DNA in which a gene unwinds and is copied into messenger RNA. So the enzymes are like RNA polymerase, and it is located in the nucleus. So in transcription, the information in the DNA of every cell is converted into small or portable RNA messages. So on the other hand, so by the way, these are the steps of transcription. So DNA is unwound at a gene. So RNA polymerase copies the DNA strand, creating a complementary messenger RNA strand. And third, messenger RNA moves from nucleus to cytoplasm and binds with a ribosome to begin translation. So this is the structure of how transcription So we have here a protein synthesis, or the major processes of protein synthesis. The first one is the transcription, in which from DNA make a new copy of messenger RNA. On the other hand, in translation, from messenger RNA will make a new copy of a proteins. So these are let's have first the differences between the DNA versus the RNA. So let's know about the difference between the two of them. Then we have here in the sugar, the deoxyribose for DNA and ribose for RNA. Also in nitrogen bases are slightly different. For DNA, we have here ATCG, and while in the RNA is AUCG, in which it has no thymine. So however, it replaced by letter U, which is called uracil, and the shape of the DNA is a double helix, while in RNA is only have single helix, and the DNA is located in the nucleus, while RNA is located in the both nucleus and cytoplasm. So here's the structure of DNA, between the DNA and the RNA. So we have here the transcription, a first step of gene expression. So a section of DNA or a gene unwinds and is copied into messenger RNA, and its enzymes are the RNA polymerase, and it is located in the nucleus. So in transcription, the information in the DNA of every cell is converted into small a portable RNA messages. So here's the steps of transcription. The DNA is unwound at a gene. Second, RNA polymerase copies the DNA strand, creating a complementary messenger RNA strand. And lastly, messenger RNA moves from nucleus to cytoplasm and binds with the ribosome to begin translation. So here's how the transcription looks like. So in the process of transcription, we have here a letter A represents the adenine, in which it will replace by U which stands for uracil. So another example for this is that the letter G also represents as a guanine, and it replaced by letter C, which is called the cytosine. So, what would pair with the following sequence? So, from the DNA, ATT, ACC, GAT, in which it means that it is the adenine, thymine, and adenine, cytosine, guanine, adenine, and thymine. It is replaced, and from the DNA, it will make a new copy of messenger RNA, in which adenine replaced by U, which is called the uracil, thymine replaced by A as adenine, T replaced by another A called adenine, and A from DNA is replaced also by U. It stands for uracil. C is for guanine. Another C is for guanine, and guanine is for cytosine, and A adenine is for uracil, and C is for adenine. So this is what we call the process of transcription, of how the DNA became transcribed into a messenger RNA. And adenine. So the second step of gene expression, or the major processes of the protein synthesis, is that the information from the mRNA is used to make a protein. So the locations are on the ribosomes of the cytoplasm. So during the translation, these messages travel from where the DNA is in the cell nucleus to the ribosomes, where they are read to make specific proteins. So here are the steps of, or the types of RNA in translation. So the messenger RNA attaches to the ribosome at the start codon. So in both prokaryotes and eukaryotes, there are three main types of RNA: the messenger RNA, ribosomal RNA, and transfer RNA. These three types of RNA are discussed, by which mRNA accounts for just 5% of the total RNA in the cell. So messenger RNA is the most heterogeneous of the three main types of the RNA in terms of both base sequence and size. So it carries complementary genetic code copied from DNA during transcription in the form of triplets of nucleotides called the codons. So each codons, so as you can see the picture right here, so each codon specifies a particular amino acids, though one amino acid may be coded for by many different codons, and although there are 64 possible codons or triplet bases in the genetic code, only 20 of them represent amino acids. And there are also 3 stop codons, which indicate that ribosomes should cease protein generation by translation. So the second one is the tRNA, or the transfer RNA. So it carries the amino acid specific to the codon on the messenger RNA. So the transfer RNA is the smallest of the 3 types of RNA, possessing around 75 to 95 nucleotides. So transfer RNAs are essential components of translation, where their main function is that transfer of amino acids during protein synthesis. So therefore they are called transfer RNAs. So each of the amino acids has a specific tRNA that binds with it and transfer it to the growing polypeptide chain. So the tRNAs also act as adapters in the translation of the genetic sequence of messenger RNA into proteins. Thus, they are also called the adapter molecules. So, transfer RNAs also have a clover cleft structure, which is stabilized by strong hydrogen bonds between the nucleotides. So they normally contain some unusual bases in addition to the usual 4, which are formed by methylation of the usual bases, and the methyl guanine and the methyl cytosine are examples of the methylated bases. So lastly, we have also the last type of RNA is the ribosomal RNA. So our ribosomal RNAs are found in the ribosomes and account for 80% of the total RNA present in the cell. So ribosomes are composed of a large subunit called the 50S and a small subunit called the 30S. So each of which is made up of the own specific ribosomal RNA molecules. So different ribosomal RNAs present in the ribosomes include the small RNAs and large RNAs, which belong to the small and large subunits of the ribosomes respectively. So ribosomal RNAs combine with proteins and enzymes in the cytoplasm to form a ribosomes, in which it acts the site of protein synthesis. So these complex structures travel along the mRNA molecule during the translation, and it facilitates the assembly of amino acids to form a polypeptide chain. So they interact with transfer RNAs and other molecules that are crucial to protein synthesis. So for example, in bacteria, the small and large ribosomal RNAs contain about 1,500 to 3,000 nucleotides respectively, whereas in humans, they have about 1,800 to 5,000 nucleotides respectively. However, the structure and the function of ribosomes is largely similar across all of the species. So the process continues until a stop codon is reached, so no amino acids is associated with any stop codon. So here's the another pictures of process of transcription and translation. So, we have a question here. Does the central dogma always apply? So, my answer is that with the modern research, it is becoming clear that some aspects of central dogma are not entirely accurate. However, current research is not focusing on the investigating of the function of the non-coding RNA. So when we say a non-coding RNA, it is a functional RNA molecule that is transcribed from DNA, but it is not translated into a protein. So there is a possibility that a DNA cannot also be translated to into a protein. So third, it is although this does not follow the central dogma, it still has a functional role in the cell. So ever since the central dogma was first proposed in 1950s, RNA's role in the protein synthesis has been widely appreciated. So today, scientists and lay people alike know that messenger RNA is essential to the process of transcription, is that the tRNA is essential to the process of translation, and the ribosomal RNA is makes up the ribosomes in which translation takes place. So what far fewer people realize is that RNA is also responsible for numerous other tasks. So for example, within eukaryotes, non-coding RNAs help regulate gene expression and modify other types of RNA. Similarly, in prokaryotes, these molecules are involved in a wide range of processes from virulence to regulation of bacterial growth. So new forms and uses of non-coding RNAs continue to be discovered, and diverse nature of these molecules has led many researchers to believe that RNA may evolutionarily predate both DNA and protein. However, much more work remains to be completed before this theory can be conclusively confirmed or denied, as well as before scientists fully understand the diverse nature of the RNA molecule. So I think that's it for today. Thank you and God bless.